It's part 4, and we're on page 290. And we have this big overview here, a little focus on what I've just been talking about. Here is the neuron. This is part of the motor unit right here. The neuron comes down, has branches. It's going to interact with the muscle at the neuromuscular junction. We're going to be having, as noted before, calcium crosses the membrane here at the end. It's going to have little, where were these guys made? They were made in the cell body, and they slid all the way down. Right, it's really cool. And so the neurotransmitter is now being released. It's binding over here. We're getting our action potential, and look at that. It's the action potential across the muscle now is entering the T-tubule, and the T-tubule is sort of jostling the cistern of the sarcoplasmic reticulum, and the cisterns are where we have the calcium. So the calcium is now being released from the triad. Okay, so calcium is now entering, and we're going to be getting stuff to happen. They even give us a little, how would I say, up close and personal here with calcium release, just the way you like it. And then they're showing us, look at this, is that we have our little bikini in place. It's very discreet at this moment. It's covering all the binding sites. The myosin head cannot bind. And then, whoa, we're going to have calcium bind to the little clasp here called the Troponin Complex. And the Troponin Complex, once calcium, we call it salting out in biochemistry, right? And so the idea here is that you're actually changing the position of this protein molecule, as it were. Long story, but at any rate, the idea is that you're shifting it. Now the binding sites are available, and we do get binding. And then once binding happens, we get the little bending of the head. Okay, and once the head is bending like that, we get contraction. So that's the deal. And then we get to our contraction cycle, and this is where we're going to spend just a few moments going through this whole little cycle here. I've had to make a couple of adjustments. I'm a biochemist, and so I had to correct. A lot of biology books really don't have very well, how would I say, constructed biochemistry. But, okay. Here we are at the resting. Now, we're supposed to be having the orange thing overlying the opening. Okay, so this is, how shall I say, this is a malfunction there, a clothing malfunction going on right here. So you have to just imagine, that whoever the star is here has her tropomyosin in the correct location, covering her, binding sites there, and that's why we're not binding at the moment. This is the charged resting position. Okay, and we're going to get binding eventually because calcium comes in and causes the little tropomyosin to move away from the binding site. Okay, so that's the deal. Calcium comes in, we get binding, and there's no ADP here. Okay, this is a phosphorylated protein. Okay, ADP is off swimming around somewhere. Okay, so we do get binding here. So that's the reality of the situation. As soon as binding happens, it's automatic. The myosin head is going to actually change conformation and shape. It's going to bend its little head. Do you see? It's bent its head now. And that means that we have contraction, just the way you like it. There never was any ADP here, but now the phosphate comes off. Okay, so now we have an unphosphorylated protein that has bent, and these things happen, it moves and it loses any of its little baggage or stuff in its pockets or whatever you want to call it. Okay, so now we have that situation. Now, we're actually kind of stuck here unless we have good circulation. And this happens all the time, You go for your afternoon run or workout, you're in some sport, you come home for dinner, and then you finish dinner, all of the blood in your body is at your gut, and you go over by the, somewhere for your little entertainment center. You throw your leg over the arm of your chair, reducing circulation, and suddenly, whoa, you have a cramp. And so that's what this is right here. This is sort of a cramp. But it doesn't have to be. It can be just it can be just a stage in the contraction cycle. Okay. In order to get out of this, you have to have what's called relaxation. And we accomplish that by actually providing the myosin head with its actual substrate, which it wants hundreds of times more powerfully than it wants the actin. Okay, so let's add the ATP. Here's the ATP. Look at that little myosin head rush right off of the actin and grab its little ATP just the way it loves it. Okay, so it's in love with that ATP right there. Okay, and as soon as it grabs its little ATP, because it's ATPase, it does a hydrolysis. It basically digests that ATP right down, and the ADP goes away, and we have phosphorylation, and we're back to our resting stage again. Isn't that cute? There's no ADP here, ADP left. Okay, so that's the contraction cycle, just the way you like it. Okay, and of course, our cadavers are all here. Why? Because they're not making any ATP. So this is where rigor mortis comes in, and this is where cramping can happen, etc. So there are some steps that have to be accomplished through the contraction cycle for it to work smoothly and properly with uninhibited flow through the whole system. So with that in mind, then we consider other little details here, and here they're actually showing us the arrangement of axons, of motor neurons, a motor unit. They're actually defining it for us, aren't they? Right here. A motor unit consists of one motor neuron and all of the muscle fibers that it innervates. Right. So if you're going to recruit more muscle fibers, you have to be speaking with more than one motor neuron. So one motor neuron, they show us the motor neuron. Again, we're coming, this is more information than you have, but it's coming from the ventral horn and coming out the ventral portion of your spinal cord. It's joining up with the spinal nerve. It's exiting out here, and it's on its way to the muscle now from the spinal nerve. And now this is a neuron, which is, it's a nerve, which is an organ, and it contains many neurons. In this case, they're only displaying two of them. So here's a neuron that comes down, and it branches here and over here, and it's grabbing those particular little muscle cells. And over here, motor unit number 2 comes over here and here and here, and so that's another motor unit. Sometimes they can be used individually if you only need a little bit of strength to do something. Sometimes you have to recruit all of them in order to do a major thing. Okay, but that's what a motor unit is. Okay, so coming from a fascicle, we have individual muscle cells coming out, innervated in this particular way. Here they're actually showing us a photomicrograph that is not all that high power. It's only 330, and so you can probably see this actually occurring. If you're looking for it in gross anatomy, you always look at the most proximal end of that muscle belly, and you begin to see how the nerve that serves that nerve, or that muscle rather, is splaying into all of its little branches, and it's going into the muscle. So come the day when we can go back to school and see cadavers, I'll demonstrate how nerves actually enter in muscles. That's supposed to be a normal part of our procedure. Okay, so we'll move on to the next part from here, then we'll talk about the physiology.